Good morning. I'm Sibeli. I'm the data curator for Genomes on a Tree, or GoaT database. I am unfortunately unable to be there with you, but I think you are in very good hands with Kirsting and also my other colleagues from the Tree of Life program at the Sanger Institute. If you want to have my contact, if you have questions or want to have the information presented here, you can access all that in this link below. The deck of slides that I'm using now are on tinyurl.com/goat-pag-31. So before I set up what GoaT and why GoaT, I must talk a little bit about the Earth Biogenome Project. And its goal of sequencing all eukaryotic species is being processed by close to 50 affiliated projects, which have already declared over 130,000 species as targets for genome sequencing. So this network of sequencing initiatives is very heterogeneous and includes projects ranging in scale from as few as 5 targets to over 60,000 targeted species. And also the scope of these projects are very diverse. So some of them are few endemic species from a specific geographic region, such as the Endemixit project. But projects can also aim full geographic regions or entire taxonomic groups. So one of the major challenges, given all these types and diversity of projects, is coordination between projects. First, to find the data. Second, to declare targets, and also, and especially, to minimize duplication of effort or waste of resources. So GoaT was created to serve as this main aggregator of metadata for all eukaryotic species that are targeted for genome sequencing. Then why GoaT, right? So the eukaryotic diversity is vast, but most of the available data is unevenly distributed across the taxa. And most of this data is just scattered in the literature. So GoaT was created to have a single place to look for types of data that would be useful for sequencing genomes. It includes not only direct values, but also, it's important, it has a mechanism to infer missing data. So as an example, if we know the genome size for several taxa, we can put them on the tree as directly measured values, as you can see for genome size here on the right. We then use the tree hierarchy to fill in expected values for higher ranks. So as our tree is based on NCBI taxonomy, this step can use simple functions such as taking the mean or median values of any descendant taxa. Then we can fill in any missing species values by taking the value from the closest known ancestral value. And it can come either from genus, from family, from subphylum. So the field values allow users to directly search any attribute for any taxon. But we label all values as direct or estimated to give an indication of how much confidence we have in the value presented. Because of course it's much better to retrieve a value from a close species within the same genus than from one ancestral value that has been measured back to phylum level, right? So also to help coordinate efforts across the EDP network, we take a similar approach, but then in this time we use data on intent and progress. And this is achieved by collecting 2 different types of attributes within the list. The first one is the target list, where you can label species as targets, as in the long list, but also as priority, such as other priority or family representatives. And the second type is the sequencing status that goes from sample collected all the way to assembly available on INSTC. So basically, what is GoaT? Is a data aggregator, right? It provides a centralized source of genome metadata and helps overall coordination, definition, and tracking of sequencing projects. And just all that is going on behind the scenes. From a user perspective, everything begins with the API, the application programming interface. So you can find the documentation here at go.genomehubs.org/API-docs. But in the API, it will find a set of endpoints to access the data in the search index. So for example, showing counts of records that match a query, fetching complete taxon records, or even returning a table of search results. So the API documentation is interactive, and by the link that I mentioned, you can go and start exploring, because there are lots of examples, and all you have to do to get started is to click around. But it's important to know the API is behind the scenes, but it feeds what you can see in the UI, and it feeds all the functionality and commands of a command line interface. I'll focus today on the UI. So the API endpoints, they are used to create functionalities in the web interface. So lookup to complete and to search for the taxa or the assemblies. The records page that collates everything that we know about one taxon, and also reports that can be used to quickly visualize the data and how it behaves. The types of data that are in GoaT usually fall into 4 broad categories. So either assembly and sequence repositories, metrics and assembly quality features, specialized database, such as chromosome and genome size specialized sources, and finally target lists, especially from the Earth Biogenome Project, that inform status of each species in a sequencing project. So the web interface, GoaT was built to allow quick visualization of data and query results that are available via the API endpoints. So all the data that you can see on the website is retrievable via API programmatically. I'll give you just a quick overview. The UI consists of a series of tabs on top, you'll see, with further information on how to use GoaT, or a collection of pre-generated reports and project pages, user cases, frequently asked questions, a place for with instructions on how to submit data, and also project dedicated pages where you can see summaries for EBP affiliated projects. But I guess most importantly, if you go to goat.genomehubs.org, you can see the search box. So I'll try to give a quick demo here of how the search box is used. The first thing that you have to know is that we index the data into, right now, 2 main indices. This is the taxon and the assembly. We're currently working on the sample and index. But yeah, you can choose between either of. You have the search box and a lot of buttons underneath that will help refine your query. The most important of the 2 are the result columns, because then you can select what you want to see from a search, and query builder. So if you don't know exactly what you're looking for in GoaT, you can just go there and build the combination of terms based on the autocomplete. So here's me at the GoaT page. And the easiest way is to first try something, right? So type the name of a species. You can actually click here for several examples. We have here some tutorials. But let's say that you want to see a species. So I'll just start typing, and GoaT can fill it out for me. So you'll see that they will have a tax name, the NCBI taxon ID, and then the name of the species. You'll see a summary of all of the data, of the preset columns for show, but actually you can select as many columns as there are attributes for each one of these subset categories. So mitochondrial assembly span, assembly level, all of these are assembly attributes For genome size, there are several ways that you can see the data, and according to the type of sources and target lists, you can see the long list, which is the full species wish list of a project. I'll click on this one just to give you an idea, and click update. So here is the long list, as you can see, and then each one of these acronyms is a project that has declared Canis lupus as their target. If you want to see in more detail one of the attributes, you can actually click on the attribute itself. You'll see that here we have in green and in orange. So it means that this value is actually coming also from a descendant, so probably a subspecies of Canis lupus. And here it is. So you have the genome size. You see that the value is this, but it's ranging from 2.74 to 3.46 gigabases. And probably this is all the observations of the genome size that has been done. If you want to see the source, there is one coming from a direct value. If you click on it, it will expand and it will take you to the exact value and a link out the original source of where you got the data. The descendant probably is related to a subspecies where the data is coming from. So if you click out of this and you want to know a little, here is the subspecies. If you want to know more details of all of the information that we have for Canis lupus, you can click here to view the records page. And in this page, you can see, well, a summary of what we have for the species on GoaT, link outs to see the data either in the Darwin portal, Blob Toolkit, and other sources. We have information about the lineage. We have all the synonyms that have been found. We have some results for the analysis via Blob Toolkit, but also many of the sources and values for each one of the attributes. Right? You'll see that for Canis, most of the data, actually, if not all of the data, comes from direct measurements, including the sample location, sex. Most of them are all direct. This is a species that is really complete. So family representative for Darwin Tree of Life project. But you'll see here that the ploidy is actually coming from clade. It can come from an ancestral level. Sometimes it will show as class, but if you expand, you can actually get to where this value is coming from to populate the ancestral value. So this is the first part of how to use GoaT. You can always go back to the main page by clicking on the GoaT up here. And if you want to explore and see more examples, you can either show examples here or browse the tree. I'll go now to this list of all bat families, just to show you that you can restrict the search by rank. So in this case, we're using family, but we could change to species, right? And this would result in all of the species present in the NCBI taxonomy. That's what the backbone taxonomy we use. A lot of the values you'll see, they are estimates, but if you want to, you can actually see assembly span and toggle out things that have been inferred from ancestral taxa and leave only directly measured values for assembly span. So this is a proxy for available assembly. And you can see here by clicking any of these, it will be redirected to either ENA or NCBI entry for the species. So this is the NCBI link. This is the ENA link. One thing that is very important in GoaT is that there is a full set of combinations of queries that you can make. So as an example, I'm not going to type on the search box. You can always use the query builder to try to find a field and populate with operators and restrict based on values, right? But the good thing is that on the UI itself, you can, the pre-generated reports and pre-generated queries will give you serve as templates for you to complete your searches. So for instance, if I go here to projects and I click on, let's see, Darwin Tree of Life project, you'll have a project page that is dedicated to reporting the progress and the targets of this project. So you'll link out, you'll have information about the bioproject ID for the project, the main contact information in case there is overlap and you want to contact the responsible or the data managers for a specific project. All the information is here. If you want to see the list of species, there are ways to query in the tutorials and the help tabs. It is explained on how to best do it. But for instance, if you want to see the sequencing status for the Darwin Tree of Life project, as soon as it loads, yes. So you have, this is one of the example of the plots that we can make that shows the amount of species and at which level step in the process it is. You can click on this and it will take you to the list of species based on those results. So here is a list that you can always retrieve as a TSV or many other format files. Okay, so back to projects. One important feature in these project pages is a series of plots that shows duplication or overlap in target between projects. So for instance, in the Tree of Life, Darwin Tree of Life long list, which is all of the targets, the 70,000 species, it is showing here that 13,000 actually are shared with other initiatives. So it's overall overlap across initiatives, right? But the following plots, they will specify what you can do about the duplications. For instance, this is a list of species in the target list that have been already sequenced. So if you open them, you retrieve a list with 363 species that all have already chromosome level assembly meeting the Darwin Tree of Life project criteria. And to get to this point, you can see that the combination of terms in the query can get very specific, very complex. So these plots are a way for you to visualize the data in a report format, but you can always retrieve the data in tabular format or programmatically. And again, remember that any of these searches that you can see in the UI, you can retrieve via API by just, you can actually visualize retrieve the results just by typing API /v2 because we are in the second version. And then you can see that this is computer readable format for your query. Anyways, back to the project page, then you can see here the duplication reports. This one are all the species that have been sequenced already to chromosomal level, and then Darwin Tree of Life don't need to do anymore. But there are other plots that show either species that are in progress by someone else, but you haven't started. So DITO has not started sequencing those. And then another one that is more critical and need overlap solving. are the plots that are of species that are in progress by a given project, but also others. So it's in progress multiple times. So that's the time to go and contact the project data managers or the project managers to solve conflict. There is always the possibility to also generate custom reports in these pages. So it's in a project by project case. But this is really cool because depending on what the plot is, you can expand and view. In this case, we're having just a visualization of config and scaffold N50. And each point here is one assembly for the DAO Tree of Life project. You can use this edit button to refine your query or refine the way you plot a scene. But you can also hit the select button and change from bean to point and investigate a specific assembly. And that will take you to the exact match of that point in the report. Yeah, so again, there is a lot that you can do on GoaT UI. I suggest you just go to the UI and start exploring. You can always start with the help. There will be pre-made links where you can see what's going on. There is a little bit of explanation of what the color key. Usually people don't know what attributes there are, and there are over 50 attributes for the taxon index and around the same number for the assembly index. So you can always see this in the help and the about. But if you just want to have an idea, you can see them as result columns as always. So if you want to see what do they have about assemblies in the assembly index, you just click them all and refresh, because they will show up as columns anyways after the search. So back to my presentation. I think we are almost running. I think the timing is good, but yeah, just this query buttons, they are useful to perform quick changes in the table that you see. So include and exclude descendants, include estimates to your results, show or not empty columns, and also, of course, clear the search. If you want to use the query builder, then you can always click to expand each field, and then you can perform queries based on specific operators and even restrict values. So if you wanted, for instance, to query all bat species for which there is no genome sequence available, that has a chromosome number lower than 20 and is not targeted by DAO Tree of Life, you could retrieve that. So there are many operators. The good thing is that in GoaT, if you start typing, you will see a lot happening on its own. So it's a learning curve there. But over time, I mean, we have all the information anyways also here in the about and help. But we have operators for how do you want to see if tax name, tax tree, or even the tax lineage only. Operators such as or, and, less than, equal to, not equal to, no. All of these operators are possible. I've included here some of the links so you can actually play if you have the slides. But this is the main point of this section. So GoaT enables queries across any combination of attributes, and also, I've already given the project page demo. So you can explore the results for pretty much all of the project pages for the Earth Biogenome Project Network. In the actual presentation link, you'll have some videos that you can follow along. I also have put some notes in there that you can explore. But finally, you have the possibility also of exploring the reports, right? So once you perform a query, you can click on each one of these buttons and use the pencil icon to select what you want to display in the reports. So we have the arc type reports and the donut reports to show progress. We have histograms with dates or categories. Maps are in progress, but you can see sample locations. Scatters are very useful to try to investigate pattern in the data. Of course, GoaT is interested in giving credit to the sources that it retrieves data from. So every time that you perform a query on GoaT, you can click on the sources report, and then you'll see each source with its link, and then how many values and attributes were used from each source. So you can cite on your publications. Table to summarize results are also possible. And finally, tree reports. So all of the trees are interactive, and you can click on the links and expand branches to see the data. You can click on one taxon and it leads you to the records page for that taxon, or you can click and hold and it will expand the branch of the tree for you. There is also a way to plot any type of the metadata along the trees. So GoaT data can be previewed in reports or downloaded for detailed analysis. And I think this is the main point that I want to make here, is that the magic of GoaT is the versatility that you can have in the queries. So I think I ran out of time, but these are the GoaT people. This is Rich, the person behind most of the code of GoaT, with Sujay, that was data manager and also responsible for a lot of the code for GoaT data. I am the curator. Max was the person that developed the GoaT command line interface. Mark Blackster gave the name and envisioned the whole thing. And Matthew and Green, they are in my team, the IT and bioinformatics infrastructure. We help with the deployment of the database. So here's the contact information. And I'd like to acknowledge everybody in the Tree of Life program, but especially Kirsten, that is hitting play on this video for me. And I think she would take any questions if there is still time. Many thanks.